All right, biology students, if you are watching this video, it's because you have been assigned a little bit of an extension into what we talked about in the last lesson. So in our last lesson, we talked about protein synthesis, which is the process of making proteins. And so I told you in that video that you might be assigned a little bit of an extension activity because I gave a very simplified overview of protein synthesis. But for some of you, we're going to have to dig a little deeper. So this isn't going to be bad. This is a quick lesson. I just need to talk to you about a couple things. So first of all, proteins are essential to life. We've said this over and over again. They're responsible for so many things that happen in the cell. So we want our cells to be able to crank out these proteins. Now, your whole genetic code, the blueprint for your body and everything in it, is made up of a language. And this language is comprised of 4 letters or 4 nitrogenous bases. So we have adenine, thymine, cytosine, guanine. And the way that they come together creates you. So these chains of nitrogenous bases are translated into proteins in the process of protein synthesis. And if you have not already watched the protein synthesis video, you need to make sure that you watch that before you watch this video. Otherwise, you will be so confused. All right, so we use the central dogma of genetics to simplify a very complex process. So protein synthesis, we said in the last video, is pretty complicated, but we can sum it up with this visual here. So we say that DNA makes RNA, which makes proteins. So the process of DNA becoming RNA is known as transcription. So there's 2 parts or 2 stages of protein synthesis. We have transcription and we have translation. So the process of DNA becoming RNA is transcription. Now remember, DNA contains the instructions for making proteins, and the DNA cannot leave the nucleus. So the mRNA is going to have to go to the DNA, make a transcript of the DNA, and then that mRNA will take that transcript to the ribosome where the next process happens, which is translation. So translation is the process of tRNA. Remember, that's the transfer RNA. Those are like little taxis that pick up and drop off amino acids. So the tRNA is going to read the mRNA, those instructions, and it's going to build amino acids that are going to link together to ultimately form a protein. Okay, so that's how protein synthesis works. Now, to this point, we have worked with DNA strands that just began with the start codon. So you didn't really have to think about it. And I did that for simplicity's sake, but that's not really how it works in the cell. In reality, it's the job of the tRNA to find the start codon, and then it has to translate the appropriate amino acid until it gets to what we call a stop codon. So the start codon will always be AUG. So the sequence of letters AUG, anytime you see that, that's going to be the start codon. That's where the tRNA knows to start reading. So when does it know to stop? Well, there's going to be stop codons as well. So I want you to look at your codon wheel on the screen. And if we look at the codon wheel, we're going to notice that AUG codes for the amino acid methionine. Methionine is always the start codon. So AUG is always going to be the start codon, and it's going to code for the amino acid methionine. Now you're also going to notice that there are stop codons as well. So if we have UGA, that's a stop codon. That's going to tell the tRNA when to stop reading. Then we have UAA, which is also a stop codon, and UAG, which is also a stop codon. So that's how the tRNA knows when to stop reading. Now, these stop codons are going to tell the tRNA that the protein is complete, and the tRNA is going to then leave and go pick up more amino acids. Okay, so that's the way the process works. Now, how's it going to look on a worksheet, a quiz, a test? So I might give you a sequence of DNA. Okay, so you see your A's, your T's, your C's, your G's. And I might ask you to model transcription. And if I ask you to do that, then you're going to take that DNA and you're going to create an mRNA strand using base pairing rules for RNA. So remember, A is going to pair with U, T is going to pair with A, and so on. All right, then what we're going to do, and this is the part that's different from the last lesson. So we are going to act as the tRNA, and we're going to start here on the left, and we're going to read from left to right, and we are going to look for the start codon AUG. Okay, so here it is. When you find it, that always codes for the amino acid methionine. Okay, and from here on out, I'm going to abbreviate these amino acids. So MET is the abbreviation for methionine. All right, and then you're going to read from there until you get a stop codon. So the next 3 letters is AGG. All right, so if I look at my chart, you read from inside to outside. So AGG codes for ARG. It's going to be the abbreviation. And if you're using a codon chart, especially in my class, the codon chart already has the abbreviations. All right, so GGA is going to code for GLY. And then we have UAG, which is going to be our stop codon. So this lets us know our protein is complete. And so when you're working on a worksheet, that's all you have to do. When you reach the stop codon, you're finished. All right, let's try one. We just tried one together. I'm going to let you do this one and then check your answers. So here is your DNA sequence. I want you to first transcribe that into mRNA. And then I want you to look for the start codon. That's going to let you know where to start. And I want you to translate that into an amino acid and continue until you reach a stop codon. So pause the video, do that, and then come back and check your answers. All right, so hopefully this is what your mRNA strand looked like when you transcribed. And then when you model translation, you're going to start by looking for the start codon, which is AUG. It's going to code for the amino acid MET. All right, and then we have AAA, which is going to code for LYS. And then GUU can be abbreviated VAL. And then AUG, this is going to be MET. And then UGA is going to be our stop codon. And so that tells you are finished. All right, we have more practice over this, so I just wanted to clarify and just kind of extend on what we talked about in our last lesson. And I will see you in the next lesson where we are going to talk about meiosis.